Assalamualaikum warahmatullahi taala wabarakatuh. My name is Muhammad Fikri Adin bin Madaud. I am second year student from Industrial Technology School, Bioprocess Division. Okay, today we will continue for the practical 4 subculture cell. Why we want to do the subculture cell? Because at certain time when the cell is grow in the culture, the space of the cell to grow will decrease. Also, the nutrient will also exhausted in the culture. So we want to transfer and dilute it and to grow it in a new cell culture to let it continue to grow. Okay. So the material we use to do the subculture is, first we will need a new culture vessel. Next, we will use the complete culture media containing 10% syrup. Next, we need to use PBS. Next, the conical tube to do the centrifuge. Next, the pre-warm media. Next, this is for we want to put waste here. This is containing the bleach. Next, lastly, the culture media. Okay. The first step what we want to do is, after we remove the cell culture from the incubator, we will visually inspect it. This is the cell culture that already grow. We will visually inspect it under the inverted microscope. Also visually inspect it under microscope. Also before that, we also visually inspect it through the phenol red in the decoloration of phenol red of the cell culture media. Phenol red will change into yellow if the condition of the cell culture, the pH of the cell culture is acidic, which means if the pH is higher than 7, the phenol red will turn into yellow. So we can observe it. If it turn into yellow, it means that the cell the cell already used the carbohydrate in the cell culture. They already use it to grow and they produce acidic. That is the byproduct of the cell, which is acidic, make the phenol red turn into yellow. So we can know that we need to subculture the cell to let it continue grow. Okay. After we examine it with the decoloration of phenol red, we examine under the inverted microscope to check for the unhealthy cell. Unhealthy cell, we can check the if the dead cell is present in the culture. Next, also we check the sign of contamination. Sign of contamination can be checked with a sign of the contamination, bacterial contamination, fungal contamination, also yeast contamination. Next, we calculate the percent confluency. Percent confluency is the percentage of the vessel covered in cells. Okay. After that, we can continue the process of the subculture. Okay. We will continue the process in the vertical flow hood, like before with the vertical flow hood. All the equipment and the surface of the vertical hood, we need to spray with the 70% alcohol, and we need to sanitize our hand before we use the vertical flow hood to reduce contamination and avoid contamination. Okay. The first step we want to do is we will remove and discard all the media into the waste container containing bleach that will kill all the remaining microbe in the medium. So we will use the pipette. This is the pipette. We take the pipette tubes. Next, we press it to the first stop, like from the previous video, and we take the media and we pour it in the waste. We take all the media to remove it. Remember to put it a second time to remove all the remaining. So after we remove all the media, After we remove all the medium from the cell culture, we can throw the pipette tubes. Okay, after that, what we do, we will briefly rinse with the sterile PBS here. We will rinse sterile PBS, and the function of sterile PBS is to remove all remaining serum in the media. Why we want to use this, remove all the serum? Because the serum contain the trypsin inhibitor. We want to remove the trypsin inhibitor because we want to use trypsin after this to detach the cell. So we use the pipette and briefly rinse all the space of the cell culture. Okay, after we briefly rinse, we add the trypsin. Okay, we will add the 3 ml trypsin. This is trypsin. We add 3 ml of trypsin into the first stop and the second stop to remove all the solution of the trypsin. Okay, after that, make sure all the trypsin touching all the surface of these vessels to make sure all the cell is detached before we incubate. After that, we will incubate this container. We incubate it for 2 minutes to make the process of detaching of the cells faster. After that, we will observe it in inverted microscope to check for the dispersion. If the cell is dispersed properly, after that, if cell is dispersed properly, we continue to the next step. The next step is we add 7 ml of complete culture media with 10% serum. The function of we add the complete culture media with 10% serum is because serum contain, like I said before, it contain the trypsin inhibitor. We want to inhibit the trypsin in the cell culture, because in the new cell culture, if trypsin is present, trypsin can strip the surface of the protein of the cell, and it will eventually kill the cell. So we don't want trypsin in our cell, so we add the serum. Also, serum can also have growth factor to the cell, so we add it 7 ml to the cells. Okay, after we add 7 ml of complete culture media, we will resuspend it. How we suspend it? We use the same pipette. We pipette it up and down. Yeah, like this. Okay, after that, we transfer transfer the solution here to the conical tube. We transfer to the conical tube. Use the pipette all of it. Okay. After we complete transfer the cell into the conical tube, we'll do the centrifugation. We will place it into the centrifuge, but we need to balance when we put in the centrifuge, which means when we put at one side, the opposite, we need to put one more conical tube with same volume. After that, we will centrifuge it at 125G for about 5 minutes. After that, we remove after 5 minutes, we take the centrifuge. We can see there are pellets and the supernatant. We remove the supernatant from the centrifuge, the cell that the media that we centrifuge. After that, we will add 5 ml of pre-warm culture media. Okay, after we centrifuge, we will remove the supernatant. Remember to use a new pipette tube. After that, we remove the supernatant, and the only thing left here is the pellet of the cells. Okay, after that, we will add 5 ml of fresh culture media. This is the fresh culture media, pre-warm media. We will add it. Okay, after we add the media, we will suspend. Like before, we suspend by preparing up and down, up and down. Okay, after we resuspend the cells, we count the cell using the hemocytometer. Okay, after we count the cell, we can transfer the cell into a new culture vessels. A new culture vessels. This is a new culture vessels. We will transfer the cell after we count the cell using the hemocytometer. The amount of the cell we transfer based on the cell count. So the appropriate amount we will transfer it to a new culture vessel. After that we transfer the new culture. After that we will bring the volume of the media using to 25 ml by using the revo media. We take it and we add until it 25 milliliter. After that we can check for the cell transfer. We can check under the inverted microscope for the cell. If the cell is transferred correctly, we can see the cell in the culture media. After that, we can incubate it 37 degrees C. That's all for the cell culture media. Thank you very much.